Okay, guys, welcome to our series of lectures, pathology of the cardiovascular system. But before then, it's good to understand the embryology, histology of these systemic or these systems before we venture into their specific pathologies. The cardiovascular system, some few facts about it. It actually is the first system to start functioning in embryo. It begins to function at the end of the third week. That's the 22nd day. And then blood flow begins during the fourth week. And actually, the entire cardiovascular system is of mesodermal origin. So those are the most important facts. Number one is the first system or organ to begin functioning. And then it begins to work at the end of the third week. That's the 22nd day. And then blood begins to flow during the fourth week. And the entire cardiovascular system is of mesodermal origin. That's very, very important. Now, let us look at the stages of the heart development. We have, most specifically, almost 5 stages of cardiac or heart development. The first one is the formation of the primary and the secondary cardiogenic field. Now, the second one can be or will be the formation of 2 endocardial tubes. So first one, we have the field is laid, the primary and the secondary cardiogenic field, and then we form 2 endocardial tubes. And then the 2 endocardial tubes will give us primary heart tube. They will join and form primary heart tube. And then the primary heart tube is going to give us the dilatations, 5 cardiac dilatations. And then the dilatations of the heart are going to form a loop, a U-shaped loop. We're going to venture into details and then we're going to see how it works. And then there's formation of a cardiac loop. And then we have the differentiation of these cardiac loops. So, and then finally, we will have septation of the heart. So we have 7 stages. The first stage is the development of the formation of the primary and the secondary heart tube, or the cardiogenic fields. And then we have the formation of 2 endocardial tubes. We have formation of the primary heart tube. Actually, the 2 endocardial tubes are going to join together, unite together to give us the primary heart tube. Then we have formation of 5 cardiogenic or cardiac dilatations. This naive primary heart tube is going to form dilatations, as we're going to see henceforth. We're going to have stage number 5 where there is a formation of a loop. These dilatations that are from the primary heart tube is going to form a U-shaped loop. And then finally, the dilatations that we formed at number 4, they are going to differentiate into various parts of the heart, and then we're going to form septations, or the septations of the heart. Now, let us begin with number 1, the formation of the primary and secondary cardiogenic fields, and this is the process. So, at the end of the primitive streak, there lies our progenital cardiac cells. So in this epiplast, the cells migrate through the streak in the splanchnic layer of the lateral plate mesoderm, where they differentiate into islands of cardiac myoblasts. We're going to see them as this diagram can show you. We have these at the cranial end of the primitive streak. And then along that pathway is where we have the migration of the progenitor cardiac cells. They migrate cranially, where we're going to have the islands of the primary heart fields. You can see those bloody islands, or the spots that are up there. Those are the islands of the progenital cardiac cells. And then this primary heart field actually is going to give us the atria, that's the right and the left atrium. It's going to give us the left ventricle, and most part of the right ventricle is going to come from the cells that have come from the primitive streak, and they have gone up into the cranial end to give us the primary heart tube. And then the remainder of the heart that is going to come from the secondary heart field is actually the corners, cordis, the truncus arteriosus, and some part of the right ventricle is going to be derived from the secondary heart field. As you can appreciate, on our left side we have a diagram, and it's showing us the secondary heart field, and then the neurotube, and something primitive like the outflow part of the heart. Then we're going to have those are a few diagrams explaining briefly how the splanchnic mesoderm or the splanchnic mesodermal cells migrate to the cranial end. They give us the primary heart field, they give us the secondary heart field, and you can see the migration of the neurocrest on the diagram B. The neurocrest cells are going to help us in the septation of the aorticopulmonary trunk, as we're going to see henceforth. That's the formation. We're done with the formation of the primary and the secondary heart field. Now we need to form the endocardial tubes. 2 endocardial tubes are going to form. That's the second stage. Remember, we had a horseshoe shape cardiogenic fields. These cardiogenic fields are going to develop and give us 2 endocardial tubes, one on the left and one on the right hand, on the right side of the developing embryo. You can see on the left hand side, the diagram is trying to show us the 2 endocardial tubes that have developed from the horseshoe shaped primary and secondary cardiogenic fields. Then there is the formation of primary heart tube. The 2 primary or the 2 endocardial tubes are going actually to join and give us a primary heart tube. And this is it. And this is how we can have a diagram representing that differentiation. We have the fusion of the primary. You can actually appreciate from diagram A, we have 2 endocardial heart tubes, and then you can see diagram B giving us the fusion of the 2 endocardial heart tubes to give us the primary heart tube. And then C is a complete fusion, and we have made now a primary endocardial tube or primary heart tube. At the 4th week is when we have that formation of the single heart tube, as we've already seen, and when it has fused together to form that heart. Now we have the primary heart tube is actually fused or fixed cranially and caudally. From one end, cranially, is the arterial side, and then caudally is the venous end. Now, blood is going to pass through the cephalic horns to 2 primitive aorta. We're going to see that. And then 3 pairs of veins are going to feed the heart, or to drain the heart from the caudal side or the caudal end. And then these are 3 pairs of veins. Remember, we've said we have the arterial end and we have the venous end. The venous end has 3 pairs of veins, and then the arterial end has 2 pairs of aorta. Now, the 3 pairs of veins are actually named as 2 common cardinal veins, we have 2 vitelline veins and 2 umbilical veins. And these are the pairs that are actually feeding the venous end of the primitive or the primary heart tube. It's now a fixed structure, and it has the arterial end and the venous end. Those are very, very important markers, and this is how you can diagrammatically appreciate it. We have we have the arterial end, where you can see the dorsal aorta, and then we have the venous end. And then we've said we have 3 pairs of veins. These are actually 6 veins. We have 2 vitelline. Remember, they are coming from the yolk sac. We have 2 umbilical veins later that are going to give oxygenated blood to the embryo. And then we're going to have 2 common cardinal veins. Those are the structures fixing the heart as the cranial and the arterial ends. We have now formation of dilatations. We have fixed our heart, our primary heart tube. We have arterial end and we have the venous end. Now you can name these dilatations, and this happens in the 21st day of development. You can see the outflow part of it. That's the arterial end, naming them from above downward or from craniocaudally. We begin from the first one. We have truncus arteriosus. That's from the arterial end. This is the first dilatations. We have the second dilatation is bulbus cordis. We have the third one, which is primary ventricle. We have the fourth one, which is the primary atrium. We have the sinus venosus, or the venous end. Remember, we said it's fixed on the outflow end of the heart is the arterial end, and it has 2 primitive aorta, and we have 6 veins, 3 pairs of veins that are forming the inflow part of the heart, and these are the structures holding the heart strong. And then you can name from up down: truncus arteriosus, we have bulbus cordis, we have primitive ventricle, primitive atrium, and we have sinus venosus. So the 5th stage of formation is formation of the cardiac loop. Now these differentiations or these dilatations are going to form a U-shaped structure that is called a cardiac loop by the reason of fast development. The heart is fast or progressively developing, and then the heart tube, particularly the bulbus cordis and the primitive ventricles, have a very fast and rapid growth. And because of the limited space, it's going to because of the limited space dorsally to the pericardial cavity, this heart of ours is going to develop, and we've said already the arterial end and the venous end is actually fixed. Because of that fixation, it gradually inversinates into the pericardial cavity and forming a U-shaped loop. And this is the manner we can have the diagrammatical presentation where we have the bulbus cordis and the primitive ventricles rapidly developing, and we don't have space dorsally, and what we're going to have is a U-shaped structure because the arterial end up there at the aortic root and the venous end is a fixed, they are fixed ends. So it happens from 22nd, 23rd and 24th days in development is when we form the loops, and it's by the influence of fast growth of the bulbus cordis and the primitive ventricles. Now, we had formed the 5 dilatations: truncus arteriosus, bulbus cordis, primitive ventricles, primitive atrium, sinus venosus. Immediately after the formation of the cardiac loop, we need to just know how they develop and differentiate into. In a nutshell, the truncus arteriosus, that is more cranial, that is on the arterial end is going to give us ascending aorta and pulmonary trunk. This is achieved through the neurocrest cells that migrate towards the truncus arteriosus and help in the septation of truncus arteriosus. We are going to see the formation of ascending aorta and pulmonary trunk, but it differentiates in adult derivative. We have the truncus arteriosus in embryology, and then it's the later structure that is going to give us ascending aorta and pulmonary trunk. The second dilatation that we need to differentiate and give us structures in adult derivatives is the bulbous cordis. I'm from cranial going caudally. The bulbous cordis, smooth upper part of the right ventricle, that's the outflow part, you can say, the conus arteriosus, the outflow part of the right ventricle is coming from the bulbous cordis. We have the other smooth part of the left ventricle, or the aortic vestibule. It is also coming from the bulbous cordis. Now, we have the smooth part and the trabeculated part of the ventricles and the atrium. The ventricles, sorry. So we have the smooth part of the ventricles and we have the trabeculated parts of the ventricles. Now, the smooth parts of the ventricles, whether right or left, it's coming from the bulbous cordis. Now, some writers can say it's ventricular proper. It's actually the trabeculated part of the heart. It is coming from the primitive ventricle. The two structures, the bulbous cordis and the primitive ventricles, are the ones that grow rapidly, and they are the ones that influence the looping of the heart. And then we have the primitive atrium, which is coming from which is going to give us trabeculated part of the right atrium and the left atrium. Then finally we have the sinus cordis, that is going to give us smooth part of the right atrium, coronary sinus, and the oblique vein of the left atrium. So those are briefly the differentiating structures in embryo. And in the adult heart, this is what you're going to see. And briefly, the diagram that you are seeing on the left side, you can see the truncus arteriosus. That's the one that is going to give us the aorta and the pulmonary trunk. You have the conus cordis, the smooth parts of the ventricular parts, or the outflow parts of the ventricles. And then we have the primitive ventricles. Those are the structures that are going to give us the left and the right ventricles, the trabeculated part, or you can say right ventricle proper or the left ventricle proper. So the smooth part coming from the bulbous cordis, they are the outflows of these ventricles. And then we're going to have another structure that you can see up there is the primitive left atrium that is not dissected open. Now, in adult heart, this is what you're going to appreciate. We've tried to get this diagram from Netter's Atlas, and you can actually appreciate that the bulbous cordis has given us the smooth upper part of the right ventricle, or the conus arteriosus. And this is the right ventricle that has been dissected open. So you can see the conus arteriosus, or the outflow part of the right ventricle, is the conus arteriosus. The origin, bulbous cordis. Then you can actually easily appreciate the trabeculated part of it. You can see where we are having the chordae tendinae, where we having the anterior papillary muscle, posterior papillary muscle. That all place is trabeculated. Is the rough part of it, the dissected part that is dissected and cut open, the flap that is open this direction, is the one that has developed from the primitive ventricle. That's very important. We can go to the left ventricle, and quickly you can appreciate if it's cut open, then you can appreciate the trabeculated part of it. The cut open flap is the one that is coming from the primitive ventricle in embryo, and in adults is the one that has trabeculations. And then the aortic vestibule is not well appreciated there, but it's the outflow part of the left ventricle. Quickly, we can now move to septation of the heart. The septation of the heart is when the primitive heart tube is going to have landmarks, is going to have the lumen cut into many chambers. That's what we call septation, because the primitive heart tube is a single lumen. All the heart that is flowing into the heart actually mixes with deoxygenated blood and oxygenated blood from the placenta. But what's going to happen during embryo, there is preferential blood flow. There is what we call preferential blood flow, whereby oxygenated blood is preferentially directed to aorta, and deoxygenated blood is preferentially directed to aorta as well, but the oxygenated one is preferentially directed to the tissues, and the deoxygenated is directed to the placenta for oxygenation. Then the primitive heart tube has a single lumen. This lumen is partitioned into 4 definitive chambers by the formation of the 4 septa. So these septas are as follows. Number 1, we have the interarterial septum. Remember, we have the left and the right atrium, so we need to form a septum to form 2 chambers. So we are also going to have arterioventricular septum. We are also going to have interventricular septum, and finally aorticopulmonary septum. Very important to have this, because in the pathology of cardiac malformations or congenital heart diseases, most of them lie on septation of the heart. So we can have a diagram to appreciate. We have a primitive heart tube that is a single tube that has undergone formation of looping, the U-shaped structure, and then on that same time we need to have right atrium, right ventricle, and left ventricle. In that perspective, therefore, we need to have what we call septation. So in the normal adult heart, this actually what you can appreciate. We have the interventricular septum, we have the muscular part of it, and then we have the membranous part of it. We have the right and the left atrium that is more membranous than muscular. So we need to have the first septation, or the first part of it is the formation of the septum in the common atrium. So what's going to happen is septum formation may involve 2 actively growing masses of tissues that approach each other. So we have one that is growing from above, and the other is expanding from below. These are known as endocardial cushions, as you can appreciate here. In diagram A, you can see bulging structures, and then those are called ridges. Those ridges are actually the ones that form septi, and they are called endocardial cushions. So the 2 cushions, we have septum primum, the first cushion that grows from above, from the roof of the primitive atrium. it grows downwards to the atrioventricular cushion. We're going to learn about the atrioventricular cushion in the in atrioventricular septation. So this septum primum grows downward, and before it actually fuses with the atrioventricular cushion, above it breaks off to give us what we call foramen primum. So the upper part of the septum primum has broken down to give us foramen secundum, and the foramen that is almost below it before it fuses with atrioventricular cushion is the one that is called foramen primum. This is it in diagrammatic presentation. We have septum primum. It's a cushion that is developing from the roof of the atrium. Then before it fuses with the atrioventricular cushion, there is a foramina. That foramina is called foramen primum. When it almost closes or touches or fuses with the atrioventricular cushion, it breaks off from where it began to develop from, or from the roof, giving us a foramina that is called ostium secundum. And then we have the ostium secundum, the foramen above. And then the septum secundum is another cushion that is developing from on the right side of the heart of the right atrium now that we formed. So it gives us an oblique foramina that is known as foramen ovale. It's an oval oblique foramina or gateway. So we have the two cushions that we formed, and we've already said that we form that oblique structure. The septum secundum forms to the right side of the septum primum, and we've already seen that. So the diagram on the left side of it, it's actually an appreciation that later in life you're going to have a foramen ovale that is going to shunt blood from pulmonary artery to the left atrium, then left ventricle to the aorta. Then we have later in life the foramen ovale is giving us fossa ovalis. So when you cut open the right atrium, you can appreciate a depression that is known as fossa ovalis. So the limbus of fossa ovalis is the septum secundum that was acting like a valve in uterol. So those are the structures that you can appreciate there. Then there is formation of the atrioventricular septum. The two cushions that are separating the atrium and the ventricular chambers. So we have dorsal and ventral growth of two thickenings, actually known as endocardial cushions. And these are the ones that are going to give us atrioventricular septum or a V septum. We have interventricular septation, we have the membranous part, and we have the muscular part. So the muscular part is going to grow from downward, upward, and then the membranous part is going to come from the roof of the atrioventricular septum. Then the aorticopulmonary septum is going to is actually the septation of the truncus arteriosus into two. So it by the help of the neurocrest cells, this structure is going to septate to give us two lumen. The two structures that are going to develop from the truncus arteriosus is the pulmonary trunk and the ascending aorta. So in adults, there's spiral formation of that, and of course you can see that septation. And we're going to see when we move to the congenital disorders how we have the malformations of septation, and most of them actually are appreciated from this side. Thank you guys. Until the next video, hit and subscribe. We meet as we discuss the congenital cardiac disorders.